Дорогие участники, мы
So,
снова
dear participants, we are back with you, and we continue a series of interesting practical events and sessions. Now we will have a seminar which will be devoted to new paradigms in neuroscience, as to what a medical student should know, what he will not find in his textbooks. Alexey Paevsky will do it, Center of Competences of NTI, New and Mobile Sources of Energy. And also, he is editor-in-chief of a very interesting portal on neuroscience and neuronovelty rule. Alexey, are you here? Yes, but I can see myself. I can see myself. Here it is. Can you see me, friends, colleagues? Yes, we can hear and see you well enough. So let me put on the presentation. Let me guess the button and the slides. So, dear friends, good afternoon. I hope that you can see the presentation. It started already, and today, of course, it's my seminar. Unlike the previous workshop, it's not a practical one. Rather, it's a call and an example for a medical student. First of all, medical students, what is worth looking at in advance as to what he's being taught and what modern textbooks on neurology and physiology have. Here's myself, my contacts here in case someone wants to ask a question or clarify things in more details. So here are the contacts. You can use them. Or, of course, you can try to find me in Odnoklassniki, but better not. So what I would like to talk today is that by my profession, I every day write and read about science, neuroscience, and very often I do lectures, and I very often face with modern medical education. And it makes me sad. Why so? Because, well, of course, I do it in different areas, urology up to surgery. But nevertheless, when I do neurology, I then realize that the textbooks are not only obsolete, they are obsolete many times. And quite recently, by chance, I bought, I don't know why, I bought a textbook on neurophysiology, and I was surprised to see that the authors of the textbook, which was out in 2016, they have been unaware of neurocells. So what are we going to discuss today? I wouldn't even call it a seminar, a conversation. Rather, well, me and my co-author, Anna Haruzha, we decided to bring in order, first of all, in our own heads and in neurosciences, and realize that in the recent many years, a lot has changed. And presently, we will talk about books, the books we have in Russian or English worth reading for those who really want to get acquainted with the basics of neuroscience. And then I will show you some online publications and resources available and leading scientific journals in neuroscience. And then as I will bring examples. I mean, we're going to talk about the changes in understanding of the main paradigms in the neuroscience in the recent, say, 20 or 30 years, which is not reflected in the modern textbooks. And then, just as an example, we will discuss one of the paradigms to see if, for instance, you have already studied the cell structure of the nervous system, and we will look at some news and changes as to what neurons are, and in some, and just to see as to how much it has changed, and how much it is not like they write about in the textbooks. Okay, let's start. Talking about the textbooks, the more or less acceptable one in Russian is this one. It is called From Neuron to the Brain. It's another publication which was first published back in the 70s, From Neuron to the Brain. It is not the first publication, Russian language. The only thing is that this publishing house, the recent issue was in 2018. That old stereotyped publication, which is just a translation from the English publication of 2003. That's a very good level of publication, and unlike very many other textbooks, this translation was done by the top neuroscientists from Russia. Well, I can, Pavel Balaban among the translators, and really top people who has a great command of both Russian and English. And the textbook has been obsolete already in some parts, but in some quite modern. It's sort of mostly in the interest, similar to the interest of the authors. Of course, if I wrote a textbook, I think I would be mostly writing about what I'm interested in. So there's a big historical part because I'm very professional in historical developments. Talking about foreign textbooks, and if you are really interested in neuroscience and want to be in, the best thing to read is the textbooks in the English language. There are many of them. And those I will be showing. If you're interested, I have a PDF, and I can write to me directly, and I will send it to you. Here are the main ones on neuroscience. The first one is Principles of Neural Science, which was published. One of the authors is the existing L. Cantor. The second one is David Purs and the others. Neuroscience, the second one, and the third one recently issued, Principles of Neurobiology. All three are good in some parts. For instance, Principles of Neuroscience, which is also called the Bible of the Neuroscience. They provide a very good idea of how fast those neurosciences are becoming obsolete and changes. The first edition was out in 1994 and included about more than 400 or 500 pages. In 2013, I think, the fifth last edition was out, the one you say in this fifth edition. In 15 to 18 years, there had been 5 editions out, 5. And in the meantime, it has been tripled in volume. In the recent edition, Principles of Neuroscience has 1,741 pages. So with only the basics and not much else. So if you want to have the full course, and when we were preparing our course, you can use the 3 of those books. And that's a good thing to do. There are several encyclopedias in neurobiology. In Springer, one was out, and so those encyclopedia, it's 4,000 to 5,000 pages each. Just compare the level. I can also provide you the links if you need. So if someone is interested, here is for the beginners, for those who are just interested in neuroscience, what is such a course? Consisting of 34 lectures, and divided it into 6 parts, 6 levels each, six mini courses, 5 to 7 lectures in each, cell level and separate nerve connections and higher neural activities. And now we are doing the 6th mini course, which is devoted to the pathology of well, I don't think you need it all, because if but the first 5 I would highly recommend to you to look up. Yeah. So we are doing this portal, Neuronovosti, Neuronews, and where we are trying to write every day the fresh news, because in 4 years of our work, we've been having each fresh piece every day. So I would advise and recommend also to read, not because it's ours, but because it really provides you the frontiers of the neurosciences. What's new? There are so many neuroscience journals which are out and issued, very many of them galore. For instance, if you go to websites or Scopus, look up for those journals, you will see neuroscience that there are more than 350 journals devoted exclusively to neurosciences, and also there are some general journals which published the general news, and some special editions also devoted to neuroscience. The last science edition was special, devoted to neurodegeneration. 4 reviews, very interesting. Talking about the top purely neuroscience journals, there are also very many of them, more than 10. And I would identify 7, distinguish 7. It's 3 journals of Nature Neuroscience, and 2 review. Nature Reviews Neurowatch and Nature Reviews Neuroscience. The impact factor is even higher than Nature itself, of the Nature Review Neuroscience. Here are 3 more very good journals. The Lancet group devoted to neurology, Neuron, of Cell Press group, which is cell level and molecular level, and Brain, a journal of neurology. It's the Brain journal is made up mostly in neurology. It's not exactly clinical, but the next level of reflexes in the nerve system. And another important journal, which of Society of Neuroscience US, it's Journal of Neuroscience. And I heard you were speaking about the Society of Neuroscience, and I would myself recommend to colleagues to pay attention to 2 conferences. Of course, there are many conferences. For instance, one we're having now on neurosciences, but there are 2 top global neuroscience conferences. Well, it's worth tracking them and following them and participate. It's the conference, which is conducted by the Society for Neuroscience every year in Chicago, San Francisco, and Washington. This year, of course, because of COVID, it is it will not be conducted, and once in 2 years, it's a saturation of European Neuroscience Society, which is devoted to It is by European Association or Federation of Neurophysiological Societies with more than 10,000 participants each. If SFN is held every year, the second one is done once in 2 years. Of course, this year it was done online. Next year, it will not be held, only the year after. So there is a chance that in 2022, FAS will be conducted offline, and I will hope very much that it will, because it is easier to go to Europe than to the United States. Last year, SFM, while the authorities of the United States did not issue visas to several or to thousands of participants, and I was not given the visa, also though I had been accredited to the conference. Okay, and another, and separately, I think I would draw your attention to. Well, here are questions. The link to conferences. Well, I think I won't be able to provide it online. The first one, sfm.com, Society for Neuroscience, and the second, hence, you can look up in Google, ENS, it's .com also, or fence.org. Well, it's very easy to Google, European Neuroscience Society. Well, it's very good that you can interrupt me and put your and write your questions into the chat, and I will be able to answer right away. And also, I would like to separately draw your attention to the Frontiers Publishing House, which is a group of journals which was out quite recently, open access. We do have questions to this open access, but they are trying to be up to the level and publish very good works and growing well enough. So the Frontiers, they have 107 journals at the moment, and more than 20 of them are devoted to neuroscience. Frontiers there are a lot of Frontiers, a lot of journals in neuroscience, and the top one is Frontiers in Neuroscience. Well, actually, I would better draw your attention not only to those journals and their open access journals, but I hope that you all know how to get this access and get the link to an article if you don't have an access. If you are unaware, then
here's another question. So, are there any textbooks and journals on interface? Yes, of course, there are such. Today, Jonathan Walpole is giving a lecture today. Jonathan Walpole and his wife are the authors of the first brain-computer interface journal. So if you're interested, please give me your contacts and I will send you a link to this textbook. Now, getting back to Frontiers. What's so good about Frontiers? They have an interesting genre, which is titled A Research Topic, when several editors can say, look, guys, we are interested in the following subject. For example, augmented brain. How can we read about brain augmentation function? So our lecturer, delivered this lecture on that, and as a result, they found 150 articles on that. So they said, we are going to collect articles for this particular topic. There are several journals, Frontiers in Neuroscience, and 2 or 3 journals participate in that. So the article is published in one of the Frontiers Group journals, and at the same time, it becomes part of the collection. Following that, when this subject is closed, I mean, when the papers are no longer accepted, and so the papers are going through the peer review, and they then are collected within this topic. So the same topic, within the same frame, they issue an ebook. And for example, if we take this augmentation of brain function, there is this research topic is second or third in readership. 11,018,000 open articles. Tinnitus, the noise in ears, tinnitus ranks first. Usually the topics can attract like 5 or 10 articles, but there are some subjects in neuroscience on BCI. There are several as well. Also about music, about sexual differences in the area of brain studies. So you can find any kind of topic if you go over these links. It's very good indeed for self-education. So if neuroscience news portal could have more volunteers donating their papers, or if you could have more funds to order such papers, we would have been able to translate them into Russian and to publish as much as possible. Otherwise, we have to use this kind of general, like for example, in augmentation of brain function 30 or 40, that we are able to retell. So there's 110 articles more. Now let's go over, I think that's the main slide. Otherwise we could have wrap up this presentation, because in fact there are certain key things which are not present in all universities, but in the textbooks. This is just a general method which is touched passing in one page, but in fact it's quite simple, you know, to find this in different textbooks. Well, there is a central, peripheral, and other types of systems. There are reflexes, Pavlov, Sechenov, monosynaptic reflexes, different neurons. Neurons interact with each other through neuromediators, and that's basically all. Well, of course, I'm a bit exaggerating, but here we have 9 subjects which are in the forefront of neuroscience on the one hand. On the other hand, they are not used to the full extent. But, well, say in BCI, Glia, it's much worse. The glial level in the neurosystem, you know, for example, the 3 parts synapse, but unfortunately there's nothing like that. So this book is focused on physiology, you know, much more than on glia in neuroscience. Lowers and neuromediators are also present there. optogenetics cannot be there because it came into being somewhat later than the last edition. Neurogenesis, we do find this there, but this is not a basic textbook. But I would like to stress once again the quality of these textbooks varies to such to a great extent. Really, you can find different textbooks, especially those who have relevant education and who can teach these subjects. But of course, we are not the only medical professionals in the world. I know there are other institutions. Anyway, well, I'm not claiming to be the ultimate truth in this particular subject. There's another important thing right here. So if we talk about the first position, neuron is a cell. Even many neuroscientists tend to forget about this. We discussed it quite seriously at the conference that was in 2018. For many neurobiologists and many neurologists, neuron is a kind of a wire conducting the potential towards action. That's the basic function which is responsible for superior neural activity. Yes, we understand that the cells contain all the organelles, and some neurologists know that about different diseases, accumulation diseases. But the specific feature of the neuron that it's quite long. Dendrites are thin, and the synthesis of all organelles depend on the sum total of neurons, and people tend to forget about this, making discoveries in these areas. Quite recently, we've seen that some neurological ailments, I don't remember which in particular right now, but occur because dendrites have the so-called Optogenetics. In Russian textbooks, outpost. There is a translation. Is there a translation in Russian textbook of this term? Well, hopefully everyone knows what Golgi device is about, but it turns out that neurons have sort of branches of Golgi apparatus, the so-called outposts, external sectors of Golgi, which are supposed to send information to dendrites. If there is no such connection in the protein sectors, we may see some diseases. Well, that's a cell, of course, and the cell contains a lot of different things on which the neuron functioning depends. Transport they don't pay much attention to these transport capabilities. So in many laboratories, they revisited neuron as a cell with complicated neural processes. So it's quite an interesting subject indeed, and they deal quite extensively with this in neurology. And of course, the glia and the glial subject is another area which has been underestimated. Neuromediators is another thing which is connected to glia, because it's well known that many scientists tend to forget about the so-called contacts even neurons are connected by electric synapses, and there are the so-called slit contacts. Also, over the last 20 to 30 years, we've learned about spheroidal. The transfer which takes place beyond synapses and neuromediators. Sometimes they are glued together, and there's quite a lot of interesting things that we can witness within these substances. Well, there are many interesting things which are used in neurology from BCI. I haven't seen much about telegraphy, and this is also another sad thing. There's only one MEG center in Russia, which is really insufficient for such a big country. Optogenetics, the neuron control through lasers, bacterial sensitivity towards light, where you can turn on and off neurons using some light signals. But maybe another 2 or 3 years when optogenetics will have a direct effect on human beings, and right now we've seen just the first tests in this area. First of all, used to recover the eyesight, and so we can build bacterial bipolars or other types of cells, and this is how we can force the retina to see the light. So, and it turns out to be much more efficient compared to the electrical retina. The brain stimulation is penetrating the textbooks little by little. There are a lot of papers on brain stimulation, electrical stimulation, ESC, and so on and so forth. So far, they are not well represented in modern literature, and possibly there's a bit more deep brain stimulation is better represented in at least in the textbooks which I mentioned. Modern views on neuroplasticity, despite the fact that we've known about this phenomenon for many years, 20, maybe more years, but papers in neurology on neuroplasticity, although everyone is talking about this as like aftermath of stroke, for example, but in fact this is a subject that is barely studied, and it's a sad thing, because when we talk about neurogenesis in adults, which even back in the 60s when it was discovered, and until today, very few articles and textbooks are devoted to neurogenesis, and I haven't had many lectures about this, and I was really glad even once when the first time in my career I saw a scientific article, not in the scientific journal, but on a website of a laboratory, an article which started with a swear word referring to neurogenesis, after everybody tried to close this subject. And so one of the scientists said, "Why are you doing this?" So basically, the key subjects, I haven't mentioned other things, but these are the key subjects that are very poorly described and touched upon in the textbooks. Even the things that are well forgotten, such as reflexes, for example. We've seen this in many classical textbooks. So what is the first reflex that they study? First of all, the knee reflex, the first neuron, the second neuron, synapse connection, and so on and so forth. But what about more complicated reflexes after that? Compositional, ever more complicated reflexes are regarded further on. But what about the, for example, mononeuron reflex, which is based on one neuron, the local reflex? You can hardly find it anywhere, although it was discovered in the Kazan Medical University back in the 19th century. Now let's go over the neurons now, which the information that I've collected is a simple question like we have 86 billion neurons plus minus 8 billion. That was actually established quite accurately about 11 years ago, but far from all textbooks contain this information, although I would like to say that a majority of people have managed to learn this figure. But the second figure, the second number, the overall number of neurons in our brain, cells in our brain, this has been a debatable issue for quite a long time because, they used some other methods to calculate them. So here is an example. Until today, new classes of neurons, brake neurons, are being discovered right now, the so-called rose-hip neurons. It's a family of neurons which are responsible for special brake effects. So far, we do not quite understand their function, but a good example I can draw right here. If this neuron could serve as a brake for the car, it could brake the car and activate the brakes only when you need to stop the car at the bakeries, for example. In 1962, the neurogenesis was discovered. Joseph Altman was behind that, and there have been many debates around that. Fortunately, this has now has emerged in modern textbooks. Some Russian authors have described the neurogenesis zones they are present in all brain parts. Even the theory of emergence of new neurons has changed. It used to be viewed as cells. It's just a process that under which the cells are split into some neurons, but now it's believed that all these cells originate from glia, from radial glia. Everything comes from glia. Section. So the closing of neurogenesis caused a huge response, which showed that the neurogenesis does exist, and the Alzheimer patients, with Alzheimer patients, it was proved that neurogenesis does exist, and it helped dozens upon dozens of patients, and neurologists should be aware of that. Neuroregulation is an important method fighting this particular disease. Now let's go over, we have less than 50 minutes. Let's go over glial cells, because in many textbooks, in reality, there are very few glial cells. So let's take a look at them. So here is, for example, one of the textbooks. Let's now open.
Содержание, да? И посмотрим. Тут я почти не вижу глиальных клеток. Сейчас посмотрим. Наверное, должны быть какие-то. А вот. Свойства и функции нейроглиальных клеток им...
So the properties and functions of glial cells. 20 pages are devoted to glial cells. The functions of cells occupy 140 or 7 pages. So the functions are not particularly are not described in detail, I would say. For example, these 3-part synapses are not, I don't see them here. Can you imagine that? So that's the basic and the most up-to-date textbooks, I should say. So in fact, in reality, the glial cells, that's the third part of my workshop. Rather a popular presentation, let's call it, which is bound just to demonstrate certain things. I'm not, after teaching you, it's not a training workshop. But it's quite interesting, you know. So we have 40 minutes of a popular lecture about glia. So I'm going to talk to you about the most basic views on glia. In many textbooks for medical workers, glia is the auxiliary cell of the central and auxiliary neural system. So the central and peripheral is just, you know, nonsense. Despite the fact that glial cells were discovered prior to neurons, the idea of glial cells, about glia as it is, was advanced by this wonderful scientist, Gabriel Gustav Valentin, in the book Über den Verlauf und die letzten Enden der Nerven, about the last end of the nerves. So that the title here, for example, is famous for its figure 54, that the first representation of the neuron in history. It was precisely Gabriel Valentin who voiced this idea about the 2 parts of the brain. The one section of the brain which can be excited by certain events and non-excited. And then another scientist in 1846, he provided the definition of neuroglia, and after him, Carl Ludwig Schleyer came out with a modern concept. This is a book of from 1894, that the nerve system runs with the help of through the so-called neuroglial ensemble, I would say. The glia emerges together with the nerve system, even in the simple organisms. See here, leech neural system. You see, even there, we can see glia. So nematode is the famous experimental species with scientists, because he has only 302 neurons and 52 glial cells. It's quite interesting also to look at this diagram dating back 2017, ratio between glia and neuron in different species. It was considered that the leech, so the simpler the species, the less glial cells it has. In 2007, it was believed that the human being has 1 to 10 to 1 or 50 to 1 proportion of glia to neurons. So when I looked at these articles dating back 2006, 10 to 1 ratio, but in fact, if you look at 2009 papers, it shows that glial cells roughly, that there's about the same number of glial cells as the neurons are in the brain. It's roughly 1-to-1 ratio. So I understand where they got this proportion from. In our cortex, in the brain cortex, the proportion between neuron and glia, 15 to 1 maybe, while the it can be a reverse in some other parts of the body. By 60 billion glial cells, we have 16 billion neurons in the cerebellum. The cerebellum contains a different proportion of that. So the majority of neurons are in cerebellum, not in the cortex. That's about 16 billion glial cells. I'm not sure that it's been in all the textbooks. Now about the peripheral nerve system, we know the glial cells are present not only in, but also in the intestines, in the intestinal organs. So they carry out different functions, including moderation of peristaltics of the intestines.
They're not the only ones. The Schwann cells are those for peripheral. There are 3 types of them. There's what is usually written about in the textbooks. It's the myelinating and the perisynaptical, which covers the neuromuscle synapses and provides the transfer. So not always will you see this figure or picture of classical figure of those cells. Here is the green one cell, which is a live cell, classical membrane, and here is the myelin. And here you can see how it folds. And we also remember that there's the myelinization allows you to make the nerve impulse faster. To compare the speed, it's 1 to even less than, and the record, I think, is about 230 meters per second of those fiber. And even here in Schwann, the cells, where we can see that there's a well-studied, we now can identify new functions. They are even more interesting. In 2013 and last year, the Science article showed that they themselves participate in the perception of pain and specialized cells which set up the glial ends, the complex. Even if you kill all the neuron part, and they still will transfer pain. So some of the perception of pain is played by the Schwann cells and do participate in the perception of pain. Again, all future scientists, you should study that. I think you should take into account it in maybe countering the pain perception, and how it might become very important in understanding different pathical pain, because we don't know why it all happens. And there are some other peripheral glial neuron cells. Because they exist in central ganglia. We do have this. Glia also plays the decisive part. At least we can see that in the textbooks. It's the vector for neurons. First, it's radial glia, and then neurons use it to crawl for their own places. And then the radial glia itself transforms into different or in glial wall or whatever, in specialized cells, and I am showing it to you here what the radial cell can transform into an astrocyte or directly in, and radial glia thus is shown, and it stays in Müller glia, which turns out to be not just an auxiliary glia, but it turns out that the Müller glia is also a fiber optic. And the work of 2007 shows that that's the fiber optic which directly transforms or transfers light to the bottom of the cell. So that's a very that's a live fiber optic. So here are the pictures of Müller glia. And Bergman glia also has its own function to provide actually well, better of all, well, you have to bury, well, there's nothing more important than in neurobiology than this. And I think I would mention here about the glia of central nerve system. And the main cells are astrocytes and microglia and oligodendrocytes, and they are produced from separate. It's a separate separate thing, and less studied cells of brain at the moment. But importantly is that dendrocytes and microglia, they are of a different character. That's why macroglia and oligodendrocytes, they are identified as macroglia. The first and the most well-known astrocytes, well, Mihailin Horship provided this name, is Austro-Hungarian pathologist. So here are nice cells we can see, and the way we see it, they resemble a spider or a star, but it's not really the fact, because now we have an opportunity to visualize the whole thing. It mostly looks like a sponge. And many authors say that, but actually there are many options like that, many variants, and you can see here 11 main types of astrocytes. And in this sense, as many researchers say, of glia cells, and I would quote one of the leading glial, Alosha Semyanov, Alexey, member correspondent. Well, he's a very serious and a very good scientist in the area. That the versatility of the astrocytes is exactly how a human being differs from predators. And if we Well, it doesn't differ very much, for instance, but the versatility of astrocytes, it differs dramatically, because there are many types of cells there. And here we are coming to a transcriptomical map of astrocytes, and in 2020 there was a work published, very interesting, but so far in mouse is used. Well, the functions of astrocytes are all here. There are a big number of them, of course, not 6 pages, but those are the main ones. Certainly they support the neurons and they provide the structure. They feed neurons. They are providers of glucose, and they regulate concentration of intercellular space. And quite recently, experimentally, it was shown that they do perform a function of this cyclical barrier, and they transport they stimulate the myelinization and the recovery after traumas and injuries, and they have their own immune system, and they participate in regulating the central the astrocytes regulate in the brains and they are responsible for breathing and a number of other capacities and things. And to say that it's an auxiliary cell is something we cannot do. No way. And here's how it looks, the gematoencephalic barrier. That's why astrocyte is headache of all oncologists, because they do not allow chemotherapy into the brain. And now about breathing. Good work of 2010 by our compatriots, Alexey and Sergei Kasparov. They work in UCL, and they react and activate the neurons controlling breathing. And importantly is the participation of astrocytes in 3-part synapses. And you know that we have a lot of neuromediators. Not all of them are described in the textbook. More than 200 molecules are now known. About 200 neuromediators are well known. There are some more important, maximally involved, and the main one is glutamate. Glutamate that's 90% of all synapses of the brain. And we all know that about synapses, that even the standard dogmatic phrase, that the Dale paradigm, saying that one neuron equals to one neuromediator. Any neuron uses only one neuromediator in its work. This paradigm is already obsolete. So because there are several mediator systems and neurons. And the most important thing here to mention is that this synapses is circled or rounded by special parts of astrocytes or appendices of astrocytes that regulate the level of glutamate and not allowing it to pour in and activate the neighboring synapses. Actually, the astrocytes participate in synaptical transfer of impulses of the brain, and that's we calling them auxiliary, not of course. And another more interesting thing to mention is cells of is 4-part and non-cell matrix oligodendrocytes. It's also a separate part. It's Penfield and Ortega who were doing the work. So they perform the function of ionization of separate. In neurons of central nerve system. Unlike the Schwann neurons, we here have each of them have a lot of shapes, and which ionize up to 30 neurons. There are some oligodendrocytes which can do it up to 30, and there are others who can do only one. And you can see here this that's a very rare picture. You don't find it in every textbook. Here is how how it looks. Up to maximum is up to 10 axons, and up to 150,000 cubic meters. So that's myelin also is very difficult thing, because it's cholesterol, and of course separately standing is and I think in my perspective is the most interesting part is microglia, which was described even before discovering about Alzheimer, and was doing it. Here are the pictures they did, and of course Rio del Ortega, who was the first one. So what's microglia? The most important discovery that was made by del Ortega was that when he said, well, there's a pathogen in the brain, the cells that he called microglia, they are transformed and from similar cells to more fatter, those who are eating the pathogens. Interestingly, a little bit later than del Ortega, in Russia, in the Soviet Union, just incidentally, the microglia was discovered by Professor Paevsky. He was a pathologist, and he was studying then the brain of those who died from polio and syphilitic, and he saw the same thing that del Ortega had seen, and here he was published only on Neuronovosti portal. Those are the microglia preparations by Paevsky, our friend Armand Diaz, who then was who worked at State University, and Paevsky worked there, and he found his archive digitalized, and so we agreed to be the first to publish it in Neuro News. Very beautiful thing. Look here, microglia also performs a big number of different functions. It performs the functions of immune system factors, and neuronal plasticity and synaptic pruning. So it gets rid of a spare one when we get old, and we know that we don't find it in every textbook. So we are born with a bigger number of synapses than we do have when we're grown up. Grow up, and we and after that, we those spare synapses which are not used, they are little by little they are cut off. So look here, it's a special video that I did for you to show how a microglia of calm. You can see they split the brain in territories, and everyone goes around to see. It's a patrolling function of microglia in the brain. If something goes wrong, it activates to start eating. Well, Michael, you're absolutely right. Michael's comment was that it's a permanent process. Yes, you're absolutely right. I'm going to talk about forgetting. Well, but initially, this synoptic, that's permanent synapses, which cuts off and maximally active is it at 18, 20 years of age. But of course, it is very active in forgetting things. And we recently showed this role. And there are different ways of activity because the signaling is very different there. More often, the activity of anthracytes are measured by calcium sensor, and the literature developed. Yeah, we do have that literature. Please write to me in VK, and if you don't have this opportunity, first you should look up on those works. There's so many of them, and there's a great review on astrocytes of Alexey Semyanov and there's a textbook on Glia, Introduction to Microglia. Our compatriot wrote it. He lives in London presently. Alexey Verhatsky. He's a professor, and I would highly recommend his books to you. And if you need, I can give you a link. Okay, I can. And I can continue. Okay, thank you very much for your questions. We will have time for Q&A. So let's move on. So phagocytosis, it is important and clear when, there's some positive emotion, clear gets a signal, a chemical signal from anthracytes, and when the cells are damaged, it also sends a signal when it eats the remnants, unused remnants. So, and here you will see how microglia is eating the coli. So here's the coli, and there's no more. It has eaten it up. Well, seemingly it was sick and it didn't operate, and microglia has eaten it up. So the role of microglia is, if it is not under control, it starts eating everything around. So here is the signaling of microglia, and here's how the glia remodels the synaptic remodeling, like CCTV camera providing images of remodeling and rules our capacity of our brain and memory. So we mentioned this forgetting, and yes, this year they showed its role in destroying the long-term memory. So it was, they took a mouse and then gave an electrical current, and then it stood still, and there was a moment of it's standing still, waiting for electrical current. And then it turned out that in 35 days, the mouse gradually forgets about this electric current shock. In 35 days, it doesn't remember. It has forgotten and feels calm. And then this mouse, the next one, before the experiment, using the toxin, the microglia was killed altogether. And after that, in 35 days, she it recollects well enough about that electrical current shock. So the microglia participates in degrading memory. So microglia has expressed regenerative capacities, and it is different in well, in males. Microglia becoming active faster and slowly in females. And this apoptosis, suicidal process is weaker. And here, the recent issues of Science on neurodegeneration microglia models and participates in ruling neurodegeneration in Alzheimer and Parkinsonian diseases. So, and that's the review. It's not only one work, but several articles. I haven't had time to read yet, but besides, in the brain, there are some others which are less important, less well-known, and less studied cells, ependymocytes and tanycytes, pituocytes. More than that, all glial cells work not on their own. Brain has a very difficult system of interaction. Look at the scheme of glial interaction. What is linked to which? Well, neuron to neuron, it's clear. It might not yet discover it. It might have some context with enterocytes and also neuromediators synapses with something else, not yet clear, and metabotropic contacts, and also there are some contacts with astrocytes and ependymal cells and with nerve cells. And I think that this scheme here also is obsolete, because the new ones have mediators and interactions with each other, and much more interesting, and forms such things like glial. Well, it's all very interesting, and we have to study it more because it plays quite a huge role. And gliomes in Schwann and different in multiple sclerosis, and we can see they do play a role, the myelinated diseases, and also what destroys, and Alzheimer, and also different diseases of central nerve system and chronic pain, and so on and so forth. So we have 15 minutes to go, and we are going to, well, that's why I left some time for questions. And I have, we have a question in our chat, and I will be happy to answer your questions in any form. And if you have questions, you can ask on Glia or whatever else. And also, I would like to once again say that if someone needs materials, or the materials, some of the materials that I mentioned, you can write to me directly, and I think I will put my contacts into the chat, and you will be able to write to me in Facebook, Contacti, by emails, and I will send it all to you, because I can well give it all to you. So here are my contacts. And also another thing to mention is that I would like to call on all of you to try to follow them, the advanced science or scientific developments, because otherwise it will be difficult to achieve anything in science. And secondly, if someone, well, it's my personal wish, if someone wants, well, I mean, we suggest you join in Neuronovosti, and if you are interested in journalistics, because this can be spare platform for you, and we do have such opportunities for students, neurologists, young scientists, biologists, and we will be happy to see you join in our team. We will give you advice, we will teach you, we will educate you, and because our colleagues now can work in other institutions. Any questions up until now? Okay, good. My email is very simple. Here it is. Q&A. Well, Michael, unfortunately, as for I was not following it because it is very difficult to reach it, because unfortunately, I haven't heard anything about that. But if you really are interested, please write me a letter, and I will look up, because something might have appeared in the meantime, and I will give you an answer. Because now I myself got interested in this. What about the balancing? How do you balance between, well, of course, I have to, well, I mean, balancing between the details. Well, of course, I have to omit some of the details while presenting. And when we are writing our news, we always provide the original article, and then and needless to say, if it's a presentation or a lecture, you have to omit very many important details. When I do a lecture to whomever it is, whether it's education or scientific popular, I know that I will not be able to teach you anything. That's why I'm trying to put some bullet points. And I, for instance,
Well, actually, I always prefer to focus on two things in my lectures. Well, of course, sometimes there's no choice. You have to answer either yes or no, and that's the first thing. And then I also leave a room for another person to delve into the problem, because without such self-learning, self-education, you won't be able to master anything. Well, hopefully we will be able to complete an important book that we are doing together with Anna, where we're trying to sort of describe difficult things in a simple and a plain language. Also, the biography of scientists that work in this area. After this, we are going to, well, perhaps I will get back to writing reviews of those 7 journals, news in this area. So I'll probably resume this work end of October. Are there any other questions, colleagues, friends?